Welcome to the AJP-Heart and Circulatory Physiology video table of contents, or from the editor's desk. We have a lot of great cardiovascular science in this month's issue. More is coming online every day. We're anxious to give you a glimpse of recent papers published in the journal, so let's take a peek. Zimmer et al. Provide a review of short and long-term features of post-myocardial infarction in human hearts and animal models from the standpoint of innate immune response. They review the early phase, including migration of inflammatory cells to the infarcted area that helps repair and remove cell debris and apoptosis of damaged cardiomyocytes. They also review the late phase, which includes an increase in oxidative stress and activation of the innate immune inflammatory response in cardiomyocytes without any obvious inflammatory cells. They discuss how blunting of this innate immune cardiomyocyte response may offer new hope for the management of heart failure. Rowe provides a review summarizing a presentation given during the Countermeasures to Cardiovascular Aging Symposium that was part of the American Physiological Society Conference on Cardiovascular Aging in August of 2017. Endothelial dysfunction, a characteristic of vascular aging, is a major risk factor for age-associated cardiovascular diseases. In women, the decline in endothelial function is attenuated until menopause, whereas the rate of decline accelerates to match that seen in men. Women have a progressive impairment in endothelial function across the stages of the menopause transition, related in part to declining estradiol levels. In contrast to women, little is known about the impact of declining testosterone levels on endothelial function in men. The review sheds light on how declines in sex hormones contribute to the vascular aging process in both men and women and will inform effective sex-specific intervention strategies to preserve vascular health and prevent cardiovascular disease. Mozolewska et al. Examined the role of the renin-angiotensin system in the cardiotoxic effects of the vascular endothelial growth factor inhibited bevacizumab, BVZ, and the tyrosine kinase inhibitor sunitinib, SNT, used to treat colorectal cancer and renal cell carcinoma, respectively. The aim of the study was to determine whether prophylactic administration of renin-Ang-2 system inhibitors would attenuate the cardiotoxic effects of BVZ and SNT in a chronic in vivo murine model. The addition of RAS antagonists downregulated expression of phosphorylated P38 and BCL2-like 19 kilodalton interacting protein 3 in SNT-treated mice. RAS antagonists partially attenuated the development of BVZ or SNT-mediated cardiac dysfunction. Azra et al. Investigated the effect of a high-fat diet on stiffness and vasoconstrictor reactivity of white adipose tissue and brown adipose tissue resistance arteries, and also investigated the interconversion of both adipose depots in the setting of a high-fat diet. Despite greater receptor-independent vasoconstriction in response to KCL, vasoconstriction in response to angiotensin II was lower in normal chow brown adipose tissue compared to normal chow white adipose tissue arteries, and similar response to endothelin-1 and norepinephrine in normal chow brown adipose tissue and normal chow white adipose tissue arteries. High-fat diet tended to attenuate reactivity in arteries from both adipose depots and increased expression of adipose markers in brown adipose tissue. The authors surmised that an increase in brown adipocytes in the absence of increased brown adipose tissue mass may be a compensatory mechanism to dissipate excess energy from a high-fat diet. Yang and Krajcik hypothesized that the exogenous angiotensin II-mediated vasoconstrictor response would be greater in older adults and would be affected by inhibition of adrenoreceptor or angiotensin type II receptor pathways. They used microdialysis to show that at the control site, older adults exhibited a greater peak vasoconstrictor response compared with younger adults, which was attenuated with adrenoreceptor blockade. Young skin exhibited a vasodilation in response to lower angiotensin II doses that was inhibited with the AT2 inhibitor. These results indicate that Ang2 has a greater vasoconstrictor influence in older than in younger individuals. Finally, Sicard et al. Investigated the consequences of right coronary artery ligation in mice. RCA ligation was performed in C57 black 6J mice. RV function was confirmed 24 hours post-RCA ligation by a decrease in the tricuspid annular plane systolic excursion and right ventricular longitudinal strain analysis. RCA permanent ligation increased the RV to left ventricular area ratio. Septum hypertrophy was associated with diastolic septal flattening. During the 4-week post-RCA ligation, LV ejection fraction was preserved, yet it was associated with impaired LV diastolic parameters. This study validates the model and offers a new opportunity to study mechanisms and treatment of RV-LV dysfunction after MI. Thank you for watching the March edition of the AJP Heart and Circulatory Physiology video table of contents. Please look at the great content on our website. Remember, the content is updated continuously, so visit often and sign up for our alerts. For those of you that will be attending experimental biology in Orlando, please stop by the AJP booth and say hello. We're happy to answer any questions you might have concerning the journal or your research. We'll see you next month, and please remember to send us your best work now.